Well, welcome everyone. My name is Amy Grumbling, and I'm a program manager at the Bee Conservancy. Before we get started, I just want to wish everyone a happy belated Pollinator Week, which was last week. And also, I want to share we have a few hive keeping guidelines for this webinar. Attendees are muted by default when you join. We invite you to add your name to your profile in Zoom if you haven't yet, and feel free to drop in your pronouns. We will have a Q&A session after the presentation, so we ask that you drop your questions in the chat and we'll go through and answer as many questions as we can. And let's take a look at our agenda. We have a very packed program for today. First, I'll be providing an overview about the Bee Conservancy, and then we'll dive right into the highlights from the first year of our Be the Solution Community Science program, including analysis of our urban bee data by Professor Ben Giuliano of Baruch College. Next, we'll have a brief Q&A on the program and the observations that we collected in year one. And then we'll close out with a brief training on how you can use iNaturalist to collect your own bee observations and join our Be the Solution projects to include your data in our year two analysis. So let's start out with a quick quiz, and you can just drop your answers in the chat. How many different species of bees do you think there are on the planet? So feel free to drop those in the chat. I want to see what people think. In the entire planet, how many different species of bee do we think there are? Any guesses? Okay, we've got 40. Anybody else? Casey says 20,000. Marie says over 200,000. All right, last guess is in. Ooh, 24,705, according to a recent estimate. 400. Okay, so the answer is there's over 20,000 species of bee on the planet, and only 8 of those species are species of honeybees. And there's even a more recent study from February that suggests that the number could be as close as 26,000, like as many as 26,000. There are over 4,000 species of bee in North America, over 400 species here in New York State. And as you can see, despite the common misconception, bees are not just black and yellow. Bees come in many different colors and shapes, and about 70% of bees live underground, while about 30% live in cavities like stems and holes in logs. And unlike honeybees, which live in colonies of about 20 to 60,000, most bees live solitary lives. Bees also come in many sizes. And this is a real-life image of an eastern carpenter bee, which is the largest bee in North America and just about over an inch in size. And then on its eye is the fairy bee, the smallest bee in North America, which is around 2 millimeters long. And the fairy bee is so small that when researchers look for them, they don't necessarily look for the bee itself. Sometimes they look for the shadows against the ground because it's easier to see their shadows than the actual bee. So all of those bees in all of their shapes and sizes and colors together are a keystone species on this planet and essential to healthy food systems and ecosystems. And together they pollinate about 80% of the world's flowering plants, which is essential as plants do countless things to ensure our survival, including transforming sunlight into food and energy. And they're also fundamental to our food systems. The USDA estimates that bees contribute about $15 billion worth of crop value of more than 130 crops annually. So bees are very important. So it's also important to note that the Center for Biological Diversity posits that about 1 in every 4 North American bee species are at increasing risk of extinction. Their decline impacts the health and stability of our food and ecosystems. So why are they in decline? These are the big factors. So habitat loss is a first factor, which isn't just about developing our natural lands, but also it includes poor land management practices on the green spaces that are available. Climate change is a big factor. Shifting temperatures and extreme weather events impact blooming and hatching patterns and habitat stability. chemical pesticides, including neonicotinoids, when one neonic-coated seed has enough active agent in it to kill around 250,000 bees. Then there's invasive plants, which contribute to habitat loss because they choke out native diversity. And then pests and diseases are also impactful. So this is where the Bee Conservancy comes in. We're a New York-based national nonprofit whose mission is to protect all bees and foster thriving food and ecosystems through community-based education, research, habitat creation, and all of this in urban and underserved areas. We do this primarily in three ways. First, by building habitat with partners like the New York City Housing Authority and through our Food Justice Award, which gifts native plants to urban farmers and to community gardeners to increase biodiversity and pollination for crops. And then through education and mentorship, which includes free public events and our Be the Future Green Workforce training program. And then finally, community science, which is what brings us here today. And now I'm going to pass it over to the Bee Conservancy's Executive Director, Rebecca Louie, to talk a little bit about our Be the Solution program.
Thanks so much, Amy. Thank you everyone for being here. We're really excited to share some juicy info with you today. So at a really high level, what's community science? People also refer to it as citizen science. And simply put, community science is engaging people from all walks of life to participate in actions that support scientific endeavors. And so you can kind of think of it as like crowdsourcing what scientists alone can't gather. And in the case of the Bee Conservancy, community science is specifically the collection of bee biodiversity data. Given the numbers of bee species that Amy just was speaking about and the fact that they're so small and that they're everywhere, you can probably imagine that there's a huge dearth of comprehensive bee data out there. And what we don't know, we can't protect. So, getting back to the work we've done, one of my favorite anecdotes is a couple years ago, the tech giant Meta was going to build an AI center somewhere, and they couldn't because rare bees were found on the property. So if you're like looking at the scoreboard, it's like Mark Zuckerberg 0, bees 1, and this is really why gathering data matters. So if we go to the next slide. In addition to community science benefiting research, conservation, advocacy, and policy decisions, there's also a lot of benefit to the participants. So we have a bunch here. They include, specifically in terms of our bee events, they include improving your ID skills and getting a better understanding of your neighborhood ecology. And perhaps best of all, which is like my favorite, having fun outdoors while possibly changing the world. Now, in 2025, the Bee Conservancy launched our community science initiative, Be the Solution, which is what brings us here today. And this slide is just an overview at a glance, and we're going to dive much deeper into our findings. But ultimately, in our first year, our goal was to see what we could accomplish if we went out to train a range of participants across a range of locations to collect and upload observations to iNaturalist. So in terms of iNaturalist, we set up our tools and protocols, and first and foremost, Be the Solution is about collecting observational data through photography. And you'll probably read a ton about other types of studies that collect specimens through protocols like pan trapping. But off the bat, we knew that we wanted to create a pretty low barrier to entry approach that also minimized the impact on pollinator life. Our primary platform was iNaturalist, which has millions of global users worldwide, and they range from novices to experts. And what they do is they upload photographs of local flora and fauna for both computer and human verified species IDs. And the data we will discuss today is all from our iNaturalist data set. However, we do want to give a shout out to Bee Machine, which sort of, aside from the iNaturalist work we were doing, the Bee Conservancy team worked with entomologist Brian Spiesman from Kansas State University to help beta test and provide ongoing feedback for Bee Machine, which is a relatively new platform. It's really cool, and it's coming out of his KSU office to basically use AI to identify bees at the species level. Now, where did we go? Given the location of the Bee Conservancy team, we focused our collection events and trainings in New York City and in select areas of the Hudson Valley, which are captured in these like screenshots here, which are from iNaturalist. We created unique projects for our sites with distinct geographic boundaries to better track cohort contributions. However, we also do have a global project for Be the Solution on iNaturalist, which captures all insect and plant observations for those who are in it, which we do ask everyone to join. Now, where possible within these areas, we prioritize data collection in New York State Climate Act designated disadvantaged community, which was 17 of our 24 sites. And these areas have been identified by the advisory group Climate Justice Working Group to, quote, ensure frontline and otherwise underserved communities benefit from the state's historic transition to cleaner, greener sources of energy, reduced pollution, cleaner air, and economic opportunities. Once we were set up for location, then we focused on the people. And so for the first year of our program, as you'll see on our next slide, our goal was to work within a range of groups and settings to prioritize the training and field experience. Like how do we get people set up to just go out there and have a great time? So putting community in community science, we tapped our ecosystem of partner organizations, which includes really amazing youth programs, land stewards, educators, scientists, and of course the general public. And the goal was really to get participants with varying levels of expertise with, and frankly, interest in bees, ecology, and conservation. So training had to establish this baseline that set everyone up for success, as I mentioned. And so our trainings were conducted both in person and with webinars and in advance of each event. The trainings included instruction on installation and usage of the apps, photographing insects in the field, photo editing and upload, and of course, the context for why bees matter. Okay, now our findings, very, very exciting. So as you'll see on this slide, well, first of all, the data in this report captures observations made across 2025 through March of 2026. And during this period, Be the Solution participants added more than 13,000, 13,000 plant and insect observations. And more than half of those observations were confirmed to be research grade at the time of the creation of this report. And we did have to kind of stop at some point, otherwise the data just keeps going and growing, which is very exciting. So this is as of March. So within that data set, there were more than 1,300 research grade bee observations made, which spanned 58 unique bee species. And the most frequently observed research grade bees are captured here. And all of these images, as well as all of the bee images you're going to see throughout the rest of this presentation, were all captured by Be the Solution participants. And, note, just for like permissions reasons, we largely selected images taken by our team, but we encourage you to go peck around in iNaturalist and look at all of them because there are a ton of really, really gorgeous, gorgeous images from the 279 participants that we trained. But, in terms of these species themselves, topping the list, we have the common, oh, could you go back, please? Thanks. Thanks. We have the common eastern bumblebee, we have the western honeybee, the brown belted bumblebee, eastern carpenter bee, and lastly, the two-spotted bumblebee. But as you saw, there was a little bit of a preview on the next slide. As we mentioned, there was a total of 58 unique bee species cited by participants. So we're not going to like read you this long list, but here's a look at other research grade bees observed in our data set. And if you could just note, there are these pink ones that are highlighted. We're going to get more into the pink in a little bit. But as you'll see on the next slide, this list goes on, and fun fact, yay, because our Be the Solution global project captures all insect and plant observations made by participants across the world, we did get this one observation of the rusty-patched bumblebee from Minnesota, which is particularly notable because, as you probably know, it is on the federal endangered species list. So this was really exciting to see. Okay, but getting back to those pink highlighted bees, the Department of Environmental Conservation's New York State Wildlife Action Plan has a very specific list of species of greatest conservation need. And our data set featured 5 of these SGCNs in New York State, which is really, really exciting. So as you can see here, I'll go through each one, but within New York State alone, we saw 17 golden northern bumblebees. We saw 14 lemon cuckoo bumblebees, both of which are species of greatest conservation need. And then on the next slide, we'll see that we saw, very excitingly, 1 slender resin bee, which is a high priority species of greatest conservation need. So that's like extra, extra rare and vulnerable. Then we have 1 yellow banded bumblebee and 7 northern amber bumblebees. And again, these are all, you can see we've like noted the location of like where the bees are and where they were photographed. And it was just so exciting to see these special species within our data set as well. Moving on. Beyond the swap list, we also spent some time digging into bees in several urban agriculture sites because, as we've stated, bees are so essential to our food systems. And so here we broke out the specific list of species that we saw at each of these locations, including, highlighting in pink once again, the SGCNs. And this data is particularly interesting to us because, through the Food Justice Award, which Amy mentioned earlier, we're really exploring how building pollinator habitat can impact wild bee and other pollinator populations at urban farms and community gardens, with hopes of native habitat also boosting crop yield in the pollinating communities that do that. And last but not least, in terms of our overall insect numbers, we have more than 2,000 research-grade non-bee observations, representing more than 700 unique species of insect. And so because we love all pollinators, we did identify the top 5 non-bee pollinators observed in our data set. So you see we've got the monarch butterflies, the silver-spotted skippers, the Eurasian droneflies, the transverse banded flower flies, and the blue-winged scolid wasps. And so, just shout out to our other winged insect friends who are pollinating all the plants out there. Okay, so now it's celebrity sighting time for our conversation. The Bee Conservancy is so grateful to Ben Juliano of Baruch College for digging into our data and providing the exciting analysis that you're about to hear. A doctoral lecturer of environmental studies in the Department of Natural Sciences at Baruch College, Ben's work is concerned with the sustainable management of human-modified landscapes to promote the mutual flourishing of people and the rest of nature, particularly insects. He holds a PhD in integrative biology and an MS in agroecology from the University of Wisconsin-Madison, and it is my absolute pleasure to introduce you to Ben and his work on bees in the city.
Thank you so much, Rebecca, for that introduction. Hi, everybody. Very happy to be here and share some of the analysis that I've done. I'm very grateful to the Bee Conservancy for bringing me into this project and getting to work with this fun data set of community science data. So let's get into it. So as Rebecca mentioned, I study landscapes, especially agricultural and urban landscapes. And one question that we landscape ecologists have is how does landscape change affect biodiversity and species? And here with this data set, a particularly interesting question is whether dense urban areas can support healthy bee communities. So as a bee conservation organization based in New York and New York City in particular, we're interested in the way that urban areas affect these species that we care about. Bees and other pollinators are very mobile. So they fly around, obviously, and some of them can travel up to like several kilometers, especially those larger bodied bumblebees. And so it's not only going to be the conditions in the local habitat, like the park or the garden that we may identify them in that matters to their health and survival, but also the conditions of that broader landscape or like mosaic that they move across. So urban ecologists and pollinator scientists are very interested in how bee communities change across natural, rural, and urban areas. And I have this little diagram here from another paper showing how urban ecologists think about this urbanization gradient from a fully like natural area that has maybe minimal human impact, like a state forest or park, through like rural agricultural areas to suburban areas, and then all the way into the city center, or even in those dense urban areas like New York City, we have green space that we know is serving as habitat for pollinators. And so, because of the way this monitoring network was designed by the Bee Conservancy, a lot of the sites really span this gradient from natural to urban. And so it allows us to ask these questions and evaluate how the context of the surrounding landscape affects bee biodiversity. So I took advantage of the bee data that came from this Be the Solution project. And it starts with these 2,276 bee observations in the global data set. I narrowed that down by first removing observations outside of the New York City or and Hudson Valley area, because I wanted to, keep it focused where the targeted sampling was, and it's where we had the most observations. So then we're down to just under 2,000 observations when we take those out. So those are the sites in New York and New Jersey. And then I decided to remove honeybees because honeybees are a domesticated species. They're more like chickens or cows than they are like wild animals. There are some wild honeybee populations, but in general, we're really interested in the wild and native bee species. So we take the honeybees out. That leaves us with just over 1,000 wild bees. And then lastly, I did another set of filtering just to account for the fact that some of the observations are only to the genus level. And so I didn't want to double count any species to be conservative. I only counted a species as a new species identified to the genus level if there were no in observations with a finer scale of classification. So it's just a conservative estimate. So that ends up with I think my numbers got flipped, but that's okay. We ended up with around 1,200 observations total. And on the other side of this relationship is the land cover data. So this is going to be the information about that urban context that I was describing before. And we use two sources for this. So the USGS, the US Geological Survey, maintains a national land cover database that's updated at this point almost annually, with sort of broad categorization of land cover types, including agriculture, forest, grassland, urban, or what they call impervious surface areas. And so for the upstate locations like in the Hudson Valley, I use this. But this database in New York City is not so great because we have such fine scale heterogeneity. That USGS dataset only occurs at like a 30-meter scale. And obviously within a 30-meter block in New York City, you can have many different types of land cover. So I turned to another data set that is created by the Nature Conservancy, most recently updated in 2021, that is actually at the 6-inch scale. So it's a much finer scale of land cover data. And I combine these data sets to evaluate the urban context of these bee observations. I then sort of grouped the bee observations together, clustered by sites. So these included the monitoring sites that Be the Solution set up and did programming with, as well as some other sort of clusters that were created post hoc, where there was like lots of observations in the greater New York area that may not have been included in one of those projects. And then I was interested in the diversity that occurred across these sites, right? Because all of these observations are not completely standardized, right? They're sort of people going out, taking observations when they can. There's different levels of effort. We can't necessarily say a whole lot about the abundance, but we can look at the diversity of the bee communities. And again, because these estimates can suffer from uneven sampling effort, we can use techniques that ecologists have developed to correct for these. And in particular, I used a technique called coverage-based rarefaction and extrapolation. And I'll try to keep it to like a brief and relatively high-level explanation. But the way that these techniques work is that they use the observed patterns of species discoveries at each site to estimate what diversity would look like if all of the sites had been sampled to the same level of completeness. So for example, we could have one site, like in this example image, site 2, where we had many, many more observations, around 75 observations. And when you do that many observations, you're more likely to observe more species. We may have had other sites like the site 3 that's in this image, where maybe we had, only around 20 or so observations. But based on how quickly we pick up new species when we add observations to our data set, we can estimate how many species are likely to be in a location if we, say, observed 50 individuals at every site. So this is sort of an estimation technique that allows us to more directly do like an apples to apples comparison across sites with different effort. And so these were the numbers that I used. And then the second modification that we do is instead of just taking the pure number of species, or what we call the species richness, there's another component of diversity, of biodiversity, that ecologists acknowledge, and that is what we call the evenness, or the relative abundance of those individuals. And so we use an index called the Shannon Diversity Index to incorporate both richness and evenness to give a more balanced picture of the composition. And I have a little illustration here to show you like why we would do this. So at these two example sites of plant diversity, let's say, or floral diversity, the site on the left, site 1, has 7 species. So it has more types of flowers. But you can see that that community is really dominated by that single sort of bluish purple flower species. And so it wouldn't really be fair to say that that's the most diverse, even though it has 7 species, but its evenness is relatively low. Whereas site 2 here has fewer species, there's 5, but they're sort of distributed more evenly in the community. And so using this diversity index, we would actually end up saying that site 2 is a more diverse site than site 1. And there are other indices that ecologists might use, but Shannon index is a good one at still giving some weight to new or rare species while also incorporating that evenness value as well. So to give you our results, I looked at, again, how the relationship between urbanization, which is measured here as the pavement and building cover within a half-mile radius of the survey sites. And then we looked at that index of bee community diversity. And I used a model that didn't assume any particular shape of this relationship. And what we found was this interesting sort of hump-shaped relationship where bee diversity in this data set at least appears to peak at intermediate levels of urbanization and also in those more natural areas. So if you look at this graph on the left side, it's more natural areas, right? Low urbanization, more trees and vegetation. And you can see that there's relatively high bee diversity there. Once we get into the around 25% urbanization, we get a drop off in bee community diversity. And then it sort of climbs back up again around 60% urbanization. And then once you get really, really urban, like 75 and above, we start to see this drop off again. So moderately urbanized areas have some of the most diverse communities. Rural, suburban, and intensively paved sites had some of the lowest bee diversity. And I think this pattern is probably a real one because it's also been documented in other studies across other cities. It's more broadly known as the intermediate disturbance hypothesis. It doesn't hold in all cases, but this is the idea that sites of intermediate disturbance have sort of more diversity of habitat types, resources, etc. And so that could lead to more different like niche space for more species. And this seems to align with the pattern that we are observing in the Be the Solution data so far, which is pretty exciting. And then some key takeaways from this analysis. I think the good news here is that cities aren't uniformly bad for bees, right? So we might assume that the more dense and concrete and buildings we get in an urban area, the more harmful it is for bees. but that's not necessarily the pattern we found. And even neighborhoods that have high density can attain high levels of bee diversity. Second, urban conservation is not in vain, right? With the right plants and management, urban green spaces can punch well above their weight for some bee species. And also, I want to caveat all of this by saying that, of course, more data will paint a clearer picture. This is analysis from just 1 year of observations with the Bee the Solution project. The Bee Conservancy team is hard at work already in the field season and observation season for 2026. And I'm really excited to see if these patterns hold up to another year of data.
Thank
Thanks.
you. Thank
Yeah.
you, Ben. Yes. And thank you also for the lead in because we're getting to the close of our report summary for now. And before we go to Q&A, I will highlight where Be the Solution is going in 2026. So we learned a lot last year, and we are a very iterative team. So we've applied a lot of things to our new season. One thing of note is, as you probably saw from our list of top 5 bees observed, the most commonly observed species were all big, charismatic, easy to spot bumblebees and honeybees. And so to improve our chances of collecting observational data for smaller bees, we are experimenting with macro photography and netting and releasing this year to see how that goes and impacts the numbers. In terms of our locations, I mentioned last year we did a bunch of kind of one-off visits to a whole range of sites. And this year we are going to travel to strategic select sites multiple times, season over season, to start getting like that real longitudinal view. And we're also going to include a deeper focus on urban ag sites, so the community gardens and urban farms that are within our networks. Lastly, as always, we will continue to engage new cohorts through training and events. And yep, as Ben said, we are excited because we are already underway and in the field and collecting some really great stuff. Now, we could not have accomplished this year without the help of many, many friends and colleagues. And so here is our epic list of high fives. We'd like to first and foremost extend a special thank you to the New York Pollinator Conservation Fund through the Office of the New York State Attorney General and the New York Community Trust for funding 2 years of this work, as well as the Con Edison Foundation for supporting our early pilot development. Thank you, everyone. It takes a hive. Now I'm going to ask, we have a couple of questions in the chat. And so I'm going to ask the TBC team and Ben to turn on your cameras, and we're going to move into Q&A. And you'll also notice a new face here. So I'd like to introduce our program manager, Louise Polk, who will be leading the iNaturalist training coming up after Q&A. And she wrangled a ton of our data this past year. So big thanks to Louise for that. And I'm going to turn it over to Amy for Q&A.
Thank you, Rebecca. Thank you, Ben. It's so exciting to hear the recaps. And I hope everybody enjoyed learning about our first year. So we have a couple questions in the chat, but if you have any more questions that you haven't added to the chat, feel free to keep them going and we'll answer as many as we have time for. So first, Casey asks, does the project automatically include any bee observations in New York City, or do observers on iNaturalist have to join the project for their data to be included? So I think I'll turn this one over to Louise because she is our iNaturalist guru on the team. So Louise.
Yeah, for our Be the Solution, all the projects, you have to be a member. We're not grabbing all bee data, partially because we're trying to track how effectively we're engaging people at our events and different engagements and trying to improve on that. So being able to see hard numbers on what our participants are doing after events are really, really helpful for us. But that's a great question.
Yes, that was a wonderful question, Casey. And shortly, you will learn how to join our project. So stay tuned for that. We have a question from Ben, which is farming the reason that there's a minimum in diversity around 25% land use cover? Ben, what are your thoughts on that?
Yeah, I think that's a really astute insight. My answer would be probably, right? So we do know, and it's been documented in other cases, that uh, rural areas dominated by agriculture, particularly large scale chemical intensive monocultures, are some of the worst types of habitat for bees, because there's just like not a lot of food, and there's also like poison potentially out there for them. So it's also been shown in other cases and suggested that one reason cities might actually be a bright spot for bee conservation is that we do have more like green space that's managed for aesthetics, food production in cities, us all of those things like parks and gardens being in close proximity to humans means that often the like urban agriculture has lower pesticide use than rural agriculture. And so yeah, I think it's very plausible that those sort of large scale agricultural sites in those like low to moderate levels of urbanization um, are not good news for bees.
Thank you, Ben. Ben, great question. Ben, great answer. Um, it looks like those are all the questions we currently have in the chat, but do keep them coming throughout the rest of this webinar. We'll collect the rest of the questions and we will send responses via email for anything that's asked beyond this point. And I am very excited to turn this over to Louise to talk a little bit more about how you can help be the solution. Louise.
I was muted. Yes, I'm just going to go back to our presentation. Oh, wait a second. Sorry. Realizing I have the wrong window open. Here we go. Yeah. So now we're going back to transition on how you can help be the solution by taking photos of local pollinators and uploading to our iNaturalist projects. Just a note, we're going to be moving pretty quickly through all of this, but the session is being recorded, so you should be able to look back and pause and pay closer attention to instructions if you get confused. But hopefully it's pretty straightforward. Now let's get into our beginner's guide to pollinator identification. So the first thing we want to point out, and Amy said this at the beginning, is that not all bees are black and yellow, but also not everything that is black and yellow is a bee. We've got our fuzzy bee right here, and fuzzy is really the word to help kind of distinguish when you're looking at insects, whether it's a bee or not a bee. Not always, but most of the time, if it's got some fuzz on it, it's probably a bee. If it has the stripes that are similar to bees, but not fuzzy, kind of like you see this wasp, the shiny, and the fly and the wasp, those are not bees. Flies mimic bees and wasps, whereas so they're not closely related. And wasps actually are the ancestors to bees. So bees have evolved from wasps, and that's why they have their similar coloring. Now we've gone through that, we're going to get into my favorites, which are the bees. Starting off with bumblebees, there's approximately 50 species in North America, 250 species worldwide. They're active from April to October, specifically because of their size and hair density. They can be out a bit earlier and stay out later because they can withstand colder temperatures. Bumblebees are the only other bees other than honeybees. They're social, meaning that they live in colonies. Bumblebee colonies are between 50 and 500 members, and they're all underground. And they're able to carry more pollen than other bees because of the amount of hair on their bodies. And you can see a few of our lovely bumblebees that we took last year. These are all kind of by just putting out by visual categorization. So large dark bees are not like a family of bees, but it's more when you're out there observing, these are kind of the categories they'll fall into. So the species numbers are approximate. There's approximately 500 species in North America and approximately 4,500 species worldwide. There's a lot of different bees that fall into this category. Mining bees, leafcutter bees, mason bees, more. We've got a wool carder bee right over here. They're all solitary bees, meaning that they live and work on their own rather than in a colony like bumble and honeybees. And they have a pretty diverse nesting and feeding styles. So we have both cavity and ground nesters, as well as specialist and generalist feeders. Specialist feeders that would fit into this category are like squash bees, where they are very closely related to the timing of when the squash blossoms are in bloom, and they're really good at pollinating squash. Versus like our Eastern carpenter bee, which they're out most of the season and they can pollinate most things. So that makes them generalist pollinators. onto our small dark bees. Last two categories people usually think are bees. These categories, people don't always, when they see them, think of bees. There are a lot of different species that fall into this category. We've got sweat bees, mining bees, carpenter bees, and they are pretty diverse and range in both ground and cavity nesting styles. They're all solitary, and because of their smaller size, they're primarily active in summer and fall. So these are the guys that are kind of coming out in the last month as things have warmed up. And I think a lot of time I've heard people say they thought they were like drone ants, because the drones have wings on them, but no, they're bees. Sometimes they are ants. And then onto my favorite category of bees, because I think they're the prettiest, are green bees. All green bees are members of the sweat bee family, but not all sweat bees are green. And there's approximately 500 species in North America and 4,500 species worldwide. They're active in the summer and fall, and they're all generalist pollinators, and they're all solitary ground nesters. And a little fun fact, which some people can find a little gross, sweat bees got their name because they like to land on people and drink their sweat. So if you've got a little metallic green thing landing on you, either it's a fly or it's a little bee who's just coming by to gather some minerals and salts. And then on to honeybees, which I think are probably the most well-known bees. They're not actually native to North America. They came from Europe in the 1600s. There's about 50,000 species, 50,000 bees in a colony. So if we think back to those bumblebee colonies where they max out at about 500, honeybee colonies are much bigger. They're social, and so they work together to build, clean, nurse, defend, and forage. They communicate and bond through pheromones of the queen and each other. Honeybees are generalist pollinators. They don't specialize in specific plants. This is why they can be kind of bussed around to different farms across the country to pollinate crops. And they are active through the spring, fall, and summer, but they stay awake through the winter, clustered in the hive. They are able to be out even though they're smaller because the hive maintains a consistent temperature. So honeybees are able to go back to a hive and warm up, unlike a lot of solitary bees who have to wait for it to get warmer. Now that we've talked a little bit about what you could see, we're going to talk about how to capture them. So our biggest piece of advice is don't use your community science app, in this case iNaturalist, to take pictures. We like to use your phone's camera function, or even better, if you have a camera, taking it with a camera. Just because you can have connectivity issues in the field. We want to try to get lots of angles. And so the faster you can just keep clicking, the better chance you have of getting really valuable pictures. We're also wanting to move slower around most wildlife. When you run up to them, they're not going to take that super well. But when you kind of approach them slowly, make it clear that you're not a threat, you're more likely to capture like more details. They're less likely to run away or fly away. We also want to try to keep the camera in focus. This can kind of be remedied if you're taking a lot of pictures, you have a better chance, but you want to make sure you're keeping the camera in focus so we get valuable pictures. And I mentioned it a little bit, but we want to get as many angles as we can. Depending on the species of bee, you cannot get a species level ID if you haven't seen like a picture of their face, or you don't know what the abdomen look like, or we don't know what the wings look like, or so getting kind of a front, top, back, side, and it would be great if we could also get an under view. It's really hard to do like when you're just out, but getting as many angles as you can is really helpful. Another big piece of advice is to focus on one plant or a cluster of plants. We mentioned not kind of intimidating them and getting over to them slowly. What's even better is if you're already there and they come to you. It means that they know you're not a threat, that when you get up close with your phone to take pictures or your camera, they're not threatened by you. They're not scared of you. They're not going to fly away. It also means you're doing less kind of chasing after like one individual insect and capturing more species usually, because they like to come to you. And if you're by a lot of flowers, they will come. And then we also recommend taking a photo as soon as you see an insect. Even a faraway photo is better than nothing at all. So we recommend trying to shoot in a burst function if you don't like clicking. You can also take a video and do like screen grabs for the best shots. A note on this, though, you may lose some metadata. So kind of, if you are fine with adding all of your location data in, you can, but taking pictures in the burst function accomplishes a similar thing. Once we've gone out, you know what you're looking for, basically anything flying. We've taken our pictures. Now we are back to whittling them down, because I told you we're taking tons and tons and tons. Those do not all get uploaded to iNaturalist because they wouldn't be useful. So we're again working within our photo app. We're choosing 3 to 4 distinct angles if you have them. Sometimes I even do 5 if you've got like all 5 angles. And we're using the clearest, best shot from each angle. These are some great shots from Amy, who was speaking earlier, of a longhorned, a two-spotted longhorn bee, I think. A little bit on what you do once you've selected your photos. We want to make sure what we're trying to identify is clearly framed. If iNaturalist saw this first photo, it might identify the wasp, but it will more likely identify the orange cone flower because it's the more prominent part of the photo. So when we crop in closer, we can see, and the app will understand, like, oh, you're trying to look at this wasp, not all the flowers around it. A warning though, we don't want to crop so much that we're losing resolution or the plant-pollinator relationship. It's really helpful to know what's attracting different bee species. So trying to keep that plant-pollinator relationship is important. And also just like when you get in and when the resolution goes down, it's not really any more helpful for us for identifying. Now that you know how to select your photos, we're going to get to know iNaturalist a little bit more. Downloading iNaturalist, I'm not going to stick on this for too long. We'll be sending a link through the chat that'll bring you to iNaturalist, but you can download it from the App Store or the Google Play. The only thing I'll note here is the newest version of iNaturalist has this logo, which is green with a white bird on it. There are 2 other versions of iNaturalist that are older, which are like iNaturalist Classic and Seek. I recommend downloading the newest. It's the easiest to use, easiest to understand, and it's what our tutorial is based on. So creating an account on iNaturalist is pretty simple. If you're comfortable using like a Google account, Google email, or Apple account, you can do that. If you're not, you can just add your email and you choose the username and password. They're both pretty fast, and they don't really ask that much of you. So once you have your iNaturalist account, you want to make sure to join our project. So we had a great question earlier about whether we were taking all everyone's data, and we are not, like I said. So if you're not a member of our project, we will not be able to see all your wonderful pollinator pictures, see what's going on with the bees in your area. So we have one project that you should join, Be the Solution Global. You can look it up, and it's also sent in the chat, so you can just go straight to that. So once we have made our account, downloaded, made our account, taken our pictures, kind of whittled them down, we are now going to make our first observation. So you want to choose your best 3 to 5 clear photos of one insect to upload. And iNaturalist is going to make its initial ID off of these pictures. There's generally like a button down here, and you can choose the option. We are uploading photos, and then we're hitting this check mark once we've selected the ones we want. We then need to consolidate the photos into one observation. It's not helpful to have 5 different observations of the same bee because it's not useful for seeing what's actually there. So we want to select all the photos you want into a single observation, and then we click this bottom stack button on the left, and then we import the observation. Then we want to complete our observation, so we select the top ID suggestion. Usually, you want to go with whatever is most like, this was very obviously a common eastern bumblebee. I had a 99.2%. So it was a pretty easy, yes, I'm going to select that. But sometimes they won't give you a species level ID. Sometimes it'll just be bumblebee. Sometimes it'll be as broad as like two-winged insect, or like it can go very broad if iNaturalist isn't sure. So we're just choosing what iNaturalist is most confident in. And other people can come in and look at your observation later and help narrow it down closer. But we don't want to pick a very specific ID that's only like a 10% chance, because less people are going to be able to determine what it is then. We're adding the location if it's not added already. This is where I was mentioning losing metadata. Usually your photos will have it already, but if you don't, you just want to add the location, because a photo observation just kind of floating with no location to tie to, it doesn't say a lot. I mean, Ben's whole presentation was about how like using the location data, and if we don't have any location data on where your bee was, it's not helpful for us. And then you can kind of edit any of this other data and upload. And you want to just keep repeating and growing your observations. It's pretty simple. It can be kind of like gamified. You get emails when you find out if someone's like confirmed your ID. I love getting research grade observations, like having it like bumped up to that level. So we'd love for you guys to all join us on iNaturalist. And so we've got a few QR codes here that you can scan. We have our iNaturalist project, which we just sent before. We have Bee Machine. We didn't talk as much about it, but we're really doing some more on our end with Bee Machine, and we'd love for you to join us there. And download our report, which has all of this data that we shared with you in even more detail, so you can really get to know what we did in our first year by downloading the report. And I think those links will be sent in the chat as well. So I won't hold on this for too long. I'm going to give it another second in case anyone's scanning. And like I said, this is recorded, so you can always pause here and scan any of these. Our other thing, we want to stay in touch and we want to help you like take action with us. We'd love feedback on this webinar. We're always trying to improve and do more. So you can provide feedback for this webinar. It takes like less than 5 minutes to do. We'd also love for you to sign up for our newsletter. We don't send it out all the time. We're not filling your inbox every week. It's like maybe once a month at most. And it's stuff that we find really interesting. And we think that if you're here, you will also probably find very interesting. And then if you're able to, donations always help. This work is very important, as Rebecca and Ben and Amy, all of us were saying, and we're hoping to be able to do it for a long time, and donations help. And so I will pass it back to Amy for some final thank yous. Or not thank yous, for some goodbyes. We're saying thank you to you for coming.
And thank you to Louise for sharing that info about how everyone can join our Be the Solution Community Science Project. So again, thank you all so much for joining us. We really appreciate you and all that you do for pollinators in New York State and beyond. We hope you had a wonderful pollinator week, and we really invite you to join us to be the solution in New York State and beyond. Again, thank you all so much, and stay tuned for upcoming events. You can check out our website.